We are going to start kidneys now. This guy's got famous kidneys. So this mammal can concentrate urine more than any other mammal per body size, about 14 times that of blood osmolarity. So, and they are desert dwelling. So, but here is a kidney. This is a kidney in cross section, or we'll just call it sagittal section, right? That shows you basically the gross organization of this organ, all right? And so what I like to do when I finally get into looking at an organ, and we're going to do some physiology on it, is you can imagine we're going to zoom in and look at all of the anatomy and histology, which will help you understand how it works, okay? So the first thing you can probably see from this picture is that there's obviously two colors, right? So you got a darker color and a lighter color here. This is a fixed kidney. And if you look really closely, or if you squint your eyes, you can see that the darker color, it looks like it's striped or striated, and is made up of a bunch of tubes as we zoom in. And so the outer part of the kidney here is called the cortex. And then these darker areas in the middle here are called pyramids. The medullary pyramids, or is considered the medulla of the organ. So soft organs can have an outer area called the cortex and an inner area called the medulla. And these pyramids are called pyramids because if you look at them, they look like rounded triangles, right? Actually, they look like little hearts, actually, since we just had Valentine's Day. But you have basically a base that's facing the exterior, and then a rounded apex, if you will, that's pointing towards the inside. And you'll then notice these canals that actually empty into a space and basically drain the kidney, okay? So we're gonna zoom in and talk about kidneys here. Here is a mouse kidney from a embryo that's been sectioned and stained so that you can kind of see these features. Here is a drawing of a typical kidney here. So all kidneys are a bunch of really long tubes, skinny tubes, wrapped in connective tissue. So for a mammalian kidney, there are millions of these tubes, and they're all basically organized in the kind of the similar way, and all sandwiched next to each other. And then we wrap them really tight in connective tissue, or what I like to call saran wrap, right? And so when you look at the outside of a kidney, you have a connective tissue layer that wraps around it and then attaches it or in place, and that is called the renal capsule. And then as you go in, what you're going to basically have are a bunch of tubes that are all kind of lined up, oriented in kind of the same way. These tubes are really long, so we got to kind of bend and fold them up so that they fit in the space that occupies the kidney. And the way the tubes are organized gives you the two different tissues that you see, or the two different textures is probably a better word than tissues. The cortex and the medulla. Now in this picture, you can see there are three pyramids here you can really see well. So here's one big one, and here's another one, and this one's actually in the best orientation at this point. So let's focus on the one down here. So this one here shows you that you got cortex that's stained slightly different on the outside, and then you have this pyramid here. The base is sticking out towards the periphery, and then you got the little rounded apex of the triangle or pyramid here, and it's called a papilla, because that's what papilla means, rounded edge. And that papilla, notice the P is sticking into a cavity here, all right? And that cavity then drains to the middle of the kidney that's labeled R, all right? And R there is the renal pelvis, all right? So that collects all of the canals or cavities that drain the medullary pyramids, or what we call the renal pyramids. and that's where the urine is collected. So the urine is collected in these cavities. The cavities are going to be called a calyx. All right, so a calyx means canal, and there are what are called minor calyces and major calyces. They're like canalways. So think minor calyces meet a renal papilla. So each renal papilla you can see in this picture down here fits nicely into a minor calyx. And if you follow minor calyces forward or towards the interior, they merge into bigger canals, right? A major calyx. And then the major calyces eventually all drain into the middle of the kidney called the pelvis. And the pelvis then empties out of the kidney through the hilus into the ureter, and the ureter drains the urine that we're producing here, right, to the bladder. Everything enters and leaves the kidney through a medial depression that is called a hilus, which is a generic anatomical term for depression. So you don't have blood vessels like penetrating the kidney all over the place. You don't have tubes penetrating the kidney all over the place. The arteries, veins, the ureters, the nerves, everything enters and leaves through that medial or middle hilus. So those are kind of all the pieces, and this is kind of urine flow. So what do kidneys do? They filter blood, and the filtrate becomes urine, all right? And this filter is pretty effective. It will filter pretty much anything dissolved in plasma. Because of that, there are going to be good things that enter the urine that we don't want to remain there, right? And so one part of the job of these tubes is to filter the blood. The other part of the job is to manipulate the filtrate, tweak it, so that we can remove any nutrients that have entered it. We can add some stuff to it if we want. But what we really want to do is make sure that the rest of that long tube tweaks the urine so that what actually leaves the kidney is waste, right? And that's a really generic term, isn't it? Waste. Waste is anything in excess. And the body typically gets rid of anything in excess or stores it somewhere, right? So, for example, if your blood glucose is really, really high, right, because you have uncontrolled diabetes, then you're going to get glucose in your waste because it's in excess, right? And so if they test your urine and it has glucose in it, that's not good. That means you have very bad glucose concentrations in the blood. Typically, if you're normal and you're healthy, for the most part, all of us have our problems, but if you're relatively healthy, any nutrients that enter the filtrate are removed, right, before the filtrate leaves the renal papilla and enters these calyces. So the stuff that you end up with is going to be what we need to get rid of, right? So if you drink tons of water, you're going to have a lot of water in your urine, right? If you don't drink a lot of water, you're dehydrated, your urine's going to be really yellow and smelly, right, with minimal amounts of water. The yellow smelly stuff is the waste, right? That's what you're doing. You're cleaning the blood, and the kidneys are really good at it. So basically the way flow rate works here, an artery called the renal artery follows the ureter up and enters through the hilus, and as it comes in to the kidney, it then branches into a bunch of smaller arteries that move towards the periphery or towards the cortex. Eventually, the renal artery breaks down into the smallest arteries in the body that are called arterioles. Okay, so arterioles are the smallest arteries. And the arteriole here is going to break down into the glomerulus, which we've kind of heard before, right? Glomerulus. All right, and what a glomerulus is a ball of blood vessels, right? A ball of the smallest blood vessels in the body that are capillaries. So the capillaries that come in here, the first at least capillary network is going to be called the glomerulus. And the blood vessels branch and get smaller and smaller and smaller so that all of the glomeruli are found in the cortex. And so that's where the filtering is going to happen, right? Because remember, we're going to surround the glomerulus, as you already know, with a capsule, right? With a balloon. And all of those balloons, or the first swollen part of our tube, are found in the cortex. All right, we know that balloon drains into a long tube we call the renal tubule. I'm going to show you pictures of this. And that renal tubule, what it's going to do is it's going to be folded up inside of the medulla and eventually will end at the papilla and drain into the minor calyces. And that will deliver what is called definitive urine, the real urine, to the calyces. And that urine will be collected by major calyces into the renal pelvis and then out to the bladder. So this is kind of the flow. So cortex right now to medulla, to the papilla, right? The little nipple-shaped structures that point into a calyx. They will drain into minor calyces, which drain into major calyces, or calyx into the renal pelvis, out to the ureter. Remember, that leaves the hilus, into the bladder for storage, and then out to the exterior, right? Eventually, as the bladder fills with urine. So what we're going to do now is we're going to zoom in and talk about those tubes, right? Because that's where all of the fun physiology happens, right? So how are these tubes organized? So if I have my kidney picture, I just brought it over here, and if I blow up really high power, one little sliver of a kidney that includes a cortex and most of a pyramid, right? Then this is what I would see if I blew it up really high under a microscope. So I blew up one piece of the kidney here, and up here on top we got the cortex, and then we have one pyramid down here, one medullary pyramid. And what I want you to notice is that I've blown up two of the tubes. Now there might be hundreds of these tubes all sandwiched next to each other. They're all going to be oriented in the same way, like this. They're going to have the balloons all found at different areas within the cortex, the little swelling right there. That's called the renal corpuscle, if you remember. And that renal corpuscle is going to drain into a long tube, right? You have these tubes, you have many, many feet of these tubes within a kidney. And the tube is really long. It's hard to fit in that small area. So we're going to fold it up and then let it dive down into the medulla and loop back up, and then eventually dive back down again and drain at the end of the papilla into a minor calyx. Now the folds here have names, particularly when it's mammalian, right? Since we're going to do the mammalian kidney. The real major difference between, say, an amphibian or a reptile or a fish nephron and a mammalian nephron is this loop right here, this first loop. All what we'll do in the other vertebrates is remove the loop, okay? So the tube will be shorter, basically, right? Now what I want you to notice with mammals, you have kind of two types of these tubes. These tubes are called nephrons. You have a cortical nephron, and you have juxtamedullary nephrons. Good names. Cortical means most of that nephron is in the cortex. Can you see that? It's short, and most of it is high in the cortex. You might get a little bit of the loop that actually dives into the most superficial part of the medulla before it comes back up. But they all drain, as you can see here, down into the papilla. The juxtamedullary nephrons are longer and deeper in the cortex. Their balloons are close to the boundary of the medulla and the cortex, and they have a much longer nephron, or renal tubule. So the renal tubule dives all the way down into the medulla and comes back up, this loop, and then it will drain back down into the loop, back down into the medulla, and it will drain at the papilla. So notice the loop here, this thing I'm calling the loop, it's called the loop of Henle, is much longer in the juxtamedullary nephrons. So in a primate like us, and then most mammals, not all, but most, you have a lot more cortical nephrons than juxtamedullary nephrons. Juxtamedullary nephrons make up, maybe between 15 and 20% of the population of nephrons. Each kidney has about, I've read different numbers, but in humans, about 650, 750,000 nephrons, so over 1 million with both kidneys. You can live with one, so if you donate a kidney, you're fine. Just don't donate both, right? So this is a histological section of what that looks like. You can't see the nephrons, right? Well, at least not on low power. But what you can see is, notice the cortex, if you look at it, kind of looks grainy to me, right? And there's some white spaces, right? Those are blood vessels. And the graininess are these balloons. And then when you look at the medulla, see it's striped. It's got dark stripes in it. Those are the renal tubules, all right? The parts of the renal tubule. So the kidneys are a simple series of tubes called nephrons, or renal tubules is what us comparative anatomists call them. They run from the superficial cortex down into the medulla, and in mammals at least they make several trips into the medulla, right? This tube as it's folded up. The long continuous tubules are called uninephros tubules. That means kidney tubules, right? And the kidney tubules, they're called this because they're actually made up of two parts, the nephron and the collecting duct. All right, so a uninephros tubule is the nephron plus a collecting duct. The renal pelvis is where is a collecting cavity of urine. Filtrates from the blood, because we're filtering blood here, and all excess waste out of the blood enter the urine. The initial solution of plasma that drains or is filtered into the nephron is called primary urine. All right, and so as this urine leaves the renal corpuscle and enters the renal tubule, it's called primary urine. And that primary urine is going to pass through a real long renal tubule where it is modified. Okay, we'll talk about how we're going to modify it. That's the physiology, right? So that what you end up with at the end that drains into the papilla, from the papilla into a calyx, looks nothing like what you started with. Okay. This fluid that passes through nephrons and collecting ducts is called filtrate. All right, it's called filtrate. All right, so far so good? You're still with me. You can see that. Oh, don't you yawn on me, girl. Yawning on me. All right, you took a test. I'll be nice. Oh, welcome to my life. I'm always up at 5:30, 6. It's called good training. Wait till you have kids. Those little bastards keep you up a lot, even as they get older. I better cut that out of the lecture. All right, anyway, so internal organization of the nephron. So this is the picture I've shown you, right, before, or drawn really poorly. So you can see this is a nephron, or what we call a renal tubule, and this will connect to a collecting duct. And so the collecting duct plus that nephron is your uninephros tubule. Now, a nephron has two parts. It has the tubule, which is the longest part, and then the swelling at the top of it, right, which is found in the cortex. And that balloon is called the renal corpuscle. And basically, it's a double-layered balloon, or think of it as two, bags, however you want to think about it. And we're going to stuff into those bags a ball of blood vessels, capillaries, we call the glomerulus. Remember, this word glomerulus means ball of yarn. That's the literal Greek translation, Latin translation of it. And what I want you to notice here is, if you look at this picture, this schematic, is we have a really small artery. Remember I told you arterioles, right? We have a really small arteriole that delivers blood to that balloon, to the renal corpuscle. And it's called the afferent arteriole. Whenever you see these words afferent and efferent, afferent means going towards, AF, towards, EF means away. So afferent arterioles deliver the blood to the renal corpuscle, which means that the afferent arteriole delivers the blood into the capillary bed, right, the glomerulus. And then that blood will travel through the glomerulus and eventually will empty out of the renal corpuscle through the efferent arteriole. Now, interestingly, this is one of the few places in the body where that efferent arteriole, where an arteriole gives rise to a capillary bed, which gives rise to another arteriole, will then give rise to another set of capillaries that are called the peritubular capillaries and the vasa recta. These are two capillary beds that are surrounding the renal tubule. So this is a really interesting setup. You have an arteriole giving rise to a capillary bed, which empties into another arteriole, which gives rise to another capillary bed. And so you got two capillary beds in tandem. Do you see that? That are connected by arterioles. Typically, an arteriole in the body, outside the kidney, typically an arteriole will deliver blood to a capillary bed, and that capillary bed will be drained by the smallest vein, which is called a venule. Okay, but here we have a different setup because we're setting up a filter system. Okay. Now, the interesting thing about the glomerulus is it's really leaky. All right, it leaks plasma. It's got fenestrations in the capillaries. You guys know what the word fenestration means? So fenestrations are small holes. The way I've always described it, which always has cracked students up, but they don't forget it, it's like you took a machine gun and blew a bunch of holes in the tube, right? And so you got thousands of these really little pores that then leak blood. Or it's like hooking a hose up to a faucet, and the end of the hose you poke a bunch of holes in, and the blood leaks, right? Now the plasma leaks, but it doesn't leak anywhere. It leaks into the renal corpuscle, right? Into our bag. All right, and the bag is the collecting part. It collects the primary urine and then delivers it to the tubule, right? And then basically the tubule passes into the medulla, it twists and winds, and guess what? Every tubule we know is lined with an epithelium. And guess what? The epithelium's going to tweak. It's going to change. It's going to move stuff in and out of the urine, as you can see here, right? You got so notice functions here. Number one, that is filtration, right? So the number one function of a nephron is filtration, to filter the blood, all right, for a certain group of molecules. That means that we filter it into the renal tubule. The renal tubule does two things. It can reabsorb material, that is, take stuff out of the filtrate and put it back into the blood, because notice we have another capillary bed that's going to surround, right, the tubule, the peritubular and vasa recta capillaries. And so, for example, any glucose that filters into the renal corpuscle is going to be reabsorbed. Matter of fact, if you're normal, 100% of it. There should be no sugar in your urine. Okay? Or I could secrete things. If it doesn't enter, enough of it doesn't enter through filtration, and I still have some in the peritubular capillaries, hell, I'll just move it from the blood into the urine, and that's called secretion. All right, so if I'm adding something to the filtrate through the blood, that's not part of the glomerulus, then I'm doing secretion. What you end up with is definitive urine, or the actual urine that you want to get rid of, right, at the end of the renal tubule. And at the end of the renal tubule, I'm going to dump that urine into a collecting duct, and that collecting duct will then drain through the papilla, okay? So as you can see down here, as I've already mentioned, a nephron is this, right? This is a nephron. A nephron consists of two parts, the part that does the filtration and the part that does the reabsorption and secretion. So you have the renal corpuscle and the renal tubule. And uninephros tubules are nephrons plus their collecting ducts, okay? Number of nephrons varies among vertebrates. Mammals have millions. Look at this. This is incredible. You have 120 kilometers of tube in your two kidneys. Did you hear what I just said? 100, and that's a lot. In the organ that's the size of a little bigger than your fist. It's almost, how do you know, if you were to line all the nephrons up, they're 120 kilometers long. Lots of tube, wonderful filtering power. The nephrons, they keep your blood clean. All right. What happens if your kidneys fail? And you don't do anything about it. You die, right? You're dead, right? So yeah, if you have kidney failure, you start feeling really bad, right? And you have to do something about it. There's intervention, but really, if your kidneys are truly failing, you're gonna need to get new ones, right? Because the kidneys that we have, our artificial cleansing of the blood is pretty poor. I'm going to be honest with you, right? So you're going to need a kidney transplant. You do dialysis until you get a kidney, but dialysis is pretty poor.
Yeah.
It doesn't do a really good job. Luckily, we are pretty good at it now. You just got to find somebody a good match, and kidney transplants are quite common actually. So, as far as organ transplants go, right? So. Now, some vertebrates don't have a lot. So cyclostomes, for example, like lampreys, they only have a couple hundred. But mammals, we have a lot. Our kidneys are very good at filtering our blood. So this is a picture of the whole thing, of our functional and anatomical units of a kidney, right? So we have a nephron, which is right here, this part right here, if I'm circling it, right? And then we have a collecting duct. All right, so here's a collecting duct. So notice the second straight dive into the medulla is the collecting duct. And we have a bunch of twists and turns to our renal tubule, right? And one of them is a long loop that dives into the medulla and then comes back up and twists again and then drains into our collecting duct. So what's really cool is the tubule has kind of different histology to it. That is, the epithelium looks different. That lines it. It is just a tube. And we know that all tubes, all hollow organs are lined by an epithelium. But the epithelium looks much different in the parts. So histologically, we can actually name the regions of the renal tubule. So the first part of the renal tubule is called the PCT, the proximal convoluted tubule. Right here, right? The proximal convoluted tubule is what collects the primary urine from the renal corpuscle, and it does a really, really good job of changing the primary urine very quickly. And we're going to talk about what it does in a few minutes. But the PCT eventually drains into a loop of Henle, all right, which is called the descending limb, which dives into the medulla. And you can see it's going to go down into the medulla and make a little hairpin turn and come back up as the ascending limb. Now look at this. Here are two tubes that are really close together going in opposite directions. What do we call that? When you got two fluids moving in opposite directions, like we had in the salt glands, right? Or in the temporal exchange in the kangaroo rat. We call it countercurrent exchange, right? So this is a countercurrent exchanger. But in reality, it's what we're going to call a countercurrent multiplier, and I'll explain the difference later. We're not there yet. That's the exciting part. We're not there, right? But the loop of Henle, as it comes up, this is the ascending limb, it will then drain into the distal convoluted tubule. And notice, this is really cool. The distal convoluted tubule comes in really close contact. Do you see that? It basically touches the side of the afferent arteriole, which is delivering blood into the glomerulus, right? And that is really, really important. That is on purpose, right? That is not something that's accidental. That part of the epithelium is going to be specialized to do some sensing of what's going on in the afferent arteriole. And we're going to talk about it. It's my favorite part of the kidney. That's where a lot of really cool physiology happens. And so we'll talk about what that part of the epithelium does. But eventually the DCT drains into a collecting duct, which will take that urine down and drop it off to a calyx, right? And those calyces eventually drain into the renal pelvis. Now, notice with the loop of Henle here, I have mammals, right? It's pretty much a mammalian. Well, birds have a small one, but and some reptiles have small ones, but it's really a mammal structure. And this, what does this do? So why do mammals have this loop? This loop allows us to concentrate our urine in the kidney. Okay? So it allows us to concentrate urine in the kidney. And guess what? A little foreshadowing. Kangaroo rats have the longest loops of Henle. Therefore, they can concentrate urine better than everybody else, right? So any animal that concentrates urine very effectively, and there are others besides kangaroo rats, usually have long loops of Henle. Okay? Now, the two parts of the renal corpuscle, and I've mentioned this before, but I want to make sure I mention it now. The balloon is called Bowman's capsule, or it's called the renal capsule. But I like Bowman's capsule. That way you don't mix it up with like the renal capsule, which actually surrounds the whole, the connective tissue that surrounds the whole kidney. But we call it Bowman's capsule, and that Bowman's capsule is double layered. There are two layers to it. And then the glomerulus, which is the ball of capillaries. That's what makes up the renal corpuscle. Both are kind of equal. They are considered renal corpuscle. And then the PCT, LH, DCT are the renal tubule, and then you got the collecting duct, which makes up the rest of the uninephros tubule. So now you can actually do urine flow, right? So glomerulus to Bowman's capsule, to PCT, to the descending limb of the loop of Henle, hairpin turn, ascending limb of the loop of Henle, DCT, to the collecting duct, out of the papilla, into a minor calyx, major calyx, renal pelvis, ureter, bush. Yeah, that's funny. Toilet. Hopefully not on the floor, like my puppy. Damn, puppies. Yes?
With the glomerulus is the capillaries.
Mm-hmm.
Is that changed just because, like, do arterioles not have the fenestrations? Is that
They don't. Yeah, just capillaries. Capillaries is the only place you exchange materials between the blood and the tissue. It's a great question. So that's the other claim to fame to capillaries. Capillaries, well, here are a couple claims to fame to capillaries, and we'll talk about them again when we get to circulation. They are the smallest blood vessel. They're the most numerous blood vessel in your body, and they're the only place exchange happens. Yeah. So it's the only place you're going to find fenestrations if they're actually fenestrations.
Is there a purpose to changing, like, or a function that we know that of changing, like, it's capillaries in the glomerulus, and then we go back to an arteriole to leave the glomerulus, and then go back to the capillary?
Yeah, there is a reason for it, because we're filtering first, and that might be enough. You know what I mean? We could potentially send it out to venous circulation, but we still need a part of the blood supply and because we've already sent the blood in and made the capillary bed, we need a part of the blood supply to actually do secretion and reabsorption. So you have to create another capillary bed. Does that make sense? So if you look here, and that's a really good question. Here is your afferent arteriole, right here. Here's your glomerulus. Here's your efferent arteriole, and then it gives rise to this huge capillary bed, which is a net of capillaries surrounding the renal tubule, because that's where you got to add things and take things away from the urine. So you need blood supply to dump things back into and to take things away from. Now, and remember when I mentioned in salamanders and freshwater fish, they have huge renal corpuscles. So their renal corpuscle will be humongous and their capillary bed would be really big because they need to produce lots of dilute urine. And then in some saltwater fish, we remove the glomerulus and the renal corpuscle altogether. And you just have one capillary bed and a tubule so that the epithelium can control what enters. So there's much finer control of what actually becomes urine in that instance. So hopefully that helps make sense out of that. Great questions. Any others? All right, well, let's start and talk about the parts and start doing a little bit of physiology, right? So that's enough histology, anatomy, organization. Here are your two nephron types that you find in mammals. Notice there are two collecting ducts. The collecting ducts all look pretty much the same. But the major difference between the cortical nephrons and the juxtamedullary nephrons is that the cortical nephrons are pretty much almost all in the cortex. They only have a very small part of their loop that enters the medulla. And so they're more superficial, shallower in the cortex. Whereas the juxtamedullary, most of the renal corpuscles are found pretty close to the boundary of the medulla. And they have really long loops, so it's a much longer nephron. And notice that almost all of the loop, pretty much all of the loop, is in the medulla. Okay? That's really important because it's the medulla where we're going to concentrate urine. All right? And there's a reason for that. The collecting ducts are also a major component of the ability to dilute or concentrate urine. They are very sensitive to hormones, the collecting ducts, okay? And we'll talk about those also. Desert species have longer loops. The most amazing thing about kangaroo rats is their loop is so long that the renal papilla, they have one major renal papilla, they have one big pyramid basically, not many, pokes out of the hilus. It's so big. All right, and so their loops are so long that they basically poke out of the hilus. I'll show you a picture of it. It's pretty funny looking. All right, so which is led right into your wonderful question there about blood supply. These are a couple cool pictures. So this is an awesome image up here. So let's start with this one here. So this is an image where they have taken some kidneys out of a mammal, during dissection, and they keep the artery and the vein intact, and they just take the whole organ out. And they also have dissected out the ureter here. And so what they do is they inject dye. They inject what look like dyes, but it's really latex. It's plastic stained dye. And you basically have injected a yellow latex, and you've filled the hilus and all the calyces, right? You also, in the artery, are going to pump basically the blood. You're going to basically pump all the blood out of the kidney, because if you take this out of a live animal, for example, an animal you killed that was recently deceased, you want to get rid of the blood. You don't want any blood in there. So you basically hook up a peristaltic pump to the artery, and you push saline through this until it runs clear, right? Once it runs clear, then you're going to add another peristaltic pump to the vein, which is much wider, and you're going to pump red dye into the arteries and blue dye into the veins. Then you stop it after a certain amount of time. If you do this correctly under the right pressure, then you throw it in an oven, and the latex hardens, right? Because it's got a catalyst in it that is temperature sensitive. Then you take that kidney and you throw it in acetic acid for 72 hours, and all of the tissue is broken down, and you're left with a cast of the blood vessels. I mean, look at that. Look at all of the blood vessels. There is a ton of blood vessels that go to this. And what I want you to notice, and I should have made this a bigger picture, notice all the arteries as they come in to, this is the hilus, right? They branch and get smaller and smaller and smaller as you move towards the cortex, right? Because that's where the capillaries are, right? That's where your glomeruli are. Now, if you were able to follow them, and this is another picture where they've used a dye to show to light up the capillaries in a kidney, and they've taken a picture of it, right, in a living body, and you can see these are the arteries coming in and the branches of them. And you can see they get smaller and smaller and smaller. And notice the smallest blood vessels are way out here, right? These are all arterioles out here. They're really small guys. That's where all the glomeruli are. So that's why all of the corpuscles are in the cortex, right? It's all the organization of the blood vessels. Eventually, those capillaries, you go afferent, glomerulus, efferent, vasa recta, peritubular, they eventually end up in a venule, all right, the smallest vein, and they drain into larger and larger veins, and then the renal vein leaves, right, draining the kidneys. The kidneys have a very, very high perfusion rate, all right? If you heard the word perfusion before, that means they receive a lot of blood. They're highly vascularized, lots of blood vessels, and they're also highly perfused. Blood goes to them. All right, a lot of blood, because you're filtering the blood, right? That's the job of the kidney, is to filter blood. Now, this is a blow up and an increased power of a little section of kidney where we're out in this area. We're out in the cortex somewhere, right? Like in this area right here where I made this box. And now I zoomed in under 40x under a microscope, and you can see here is a branch of the renal artery branch, right here, and here are one of the afferent arterioles. Now, how do I know it's afferent? Because afferents are wide. See how wide that is? And here's my glomerulus, my capillary bed, basically wound up into a ball and stuck into this capsule. You can see the capsule, look at that. See, there's a light area around it? And then it drains through a much thinner efferent arteriole, and that efferent arteriole then gives rise to another capillary bed. Now, if you follow the artery up here in this picture into the cortex and zoom in, it'll look like this, right? And this is kind of two-dimensional because this is a picture of one of those casts. You can see an afferent arteriole, an efferent arteriole, and then you can see a ball of capillaries, right? The glomeruli. So this shows you blood flow, right? And that blood flow is high. Blood pressure in the afferent arteriole is high. Perfusion rate or blood flow through the afferent arteriole is high, right? And that makes a lot of sense because it's blood pressure that drives filtration. Without blood pressure, filtration stops. Okay, so you got high blood pressure, high perfusion into the glomerulus, so that blood will leak from the glomerular capillaries into Bowman's capsule. Now, there is smooth muscle around the afferent and efferent arterioles. All right, smooth muscle can be controlled. You can relax it, you can contract it. All right, now here's what's really amazing. If you want to manipulate the filtrate in a consistent way, remove all the glucose. Remove a certain amount of sodium chloride, remove a certain amount of urea. You have to have a flow rate through the nephron, right? Because think about it. Just, bear with me a minute. If you think about this. If you are pushing too much blood into the nephron, then the flow rate's going to be too fast. And the epithelium of the renal tubule is not going to be in contact with the urine for long enough, right? If it's moving too slow, then the epithelium is in contact with the urine too long, right? It might remove too much stuff from the urine. So you want this consistent flow rate. Well, how do you control flow rate of urine? Blood flow, right? You can control blood flow. And that's what this smooth muscle does. You wrap smooth muscle around the afferent and efferent arteriole. These are called sphincters, and you can control them, right? You can control these sphincters so that, let's say you're all worked up, or you have some, you get up, you start moving around, your blood pressure goes up in your body, somebody makes you really angry, whatever it is. Gribbons makes you angry learning all this stuff, your blood pressure goes up. And your blood flow to the capillaries all over your body and maybe to the glomeruli goes up. You can close those blood vessels down a little bit, right? and kind of slow blood flow because it's too fast. Or if blood flow drops, like when you go to bed, or you lay down, or you're meditating, whatever it is, right? And your perfusion rates drops, then you can open them up, right? So you can tweak just blood flow using smooth muscle sphincters. And we'll talk about some other ways you can control blood flow. So this afferent arteriole is kind of your delivery system, right, to your filter, and you can control filtration rate by controlling blood flow in the afferent arteriole. Does that make sense? Now, the actual glomerulus and the way Bowman's capsule is set up is my favorite histological part of the kidney. And, I've published several papers on glomerular filtration in salamanders, for example. So, I love this part of it. This is one of the better drawings I could find of kind of how the glomerulus and Bowman's capsule are organized. So, as I mentioned, you have an arteriole coming in and an arteriole leaving, and you have a capillary bed between the two arterioles, right? And so basically what we're going to do is we're going to shove this capillary bed into that balloon, and to do that we have to ball it up into a ball of yarn, basically, or a fist, so that it will sit in this swelling. All right, the outside of the Bowman's capsule, the actual balloon wall, if you will, is called basically the outer layer, or the serosal layer of the Bowman's capsule. As I mentioned before, Bowman's capsule has basically two layers. The outer layer is simple squamous epithelium. So if you remember in your histology section, I told you to go look at Bowman's capsule in order to see simple squamous epithelium, right, in your histology slides. So the outer layer of the balloon, which is collecting the urine, is a simple squamous epithelium with really nice tight junctions, and it basically collects the urine that's filtering out of the glomerulus. There is an inner layer of cells. So if you follow this down here, this drawing, to where the simple squamous epithelium meets the capillary edges, then notice you'll have this purple layer of cells that then start surrounding the capillaries. And these are the inner layer, or what's called the visceral layer of Bowman's capsule. It's the inner layer of the bag, if you will, the double layer. And these cells are some of the coolest cells, most specialized cells in the body. They're called podocytes. All right, they're called podocytes. All right, and that word, podocytes, right? So this word podocyte, podo means foot, cyte means cell. So they're foot cells. And they look like a really weird cell. They have a cell body and then two long processes that come off of them. And those processes wrap around the capillaries. Okay? And it's hard to see when you section through it like this, but you'll see these little humps, which are kind of the cell body, and there'll be nucleus and all the organelles, and then they'll send this really long cytoplasmic process called a pedicel. All right? That long process that comes off of it is called a pedicel, which means small foot. All right? And that's where the name podocyte comes from, because of the pedicels. It'll send off these long pedicels. So you take a really long strip of epithelium, a really long strip of cytoplasm and membrane, and then you wrap it around the blood vessel really tight, so that the wraps are really tight. And between the wraps are where I'm going to filter the blood. All right, so these podocytes are the filtering mechanism. Or if you've ever owned a filter or talked about filtration, there's something called a sieve, right? The screen that actually determines the size that you have to be filtered, right? to actually move from the blood into Bowman's capsule. If you're too big, you hit the screen, right? And you don't go. If you're small enough, you can pass through the little holes in the screen. It's the podocytes and the pedicels that produce the screen. So if I zoom in right here in this little area right here, oh, I'm out of time. This is where we'll start next time. But if I zoom in right here, these are these little feet, these little wraps. And notice they're parallel to each other. And between them is where the blood actually squeezes, or the plasma squeezes out to enter Bowman's capsule. Okay? Have you guys ever seen the movie Aliens? It's an old movie. It's an old movie. It's a great movie. No, so if you look up the original movie Alien, it was it's a great movie. But the alien, the larva of the alien looks like a big podocyte. It attaches to your face and then wraps its process around your neck. Yeah, that's exactly what a podocyte looks like.
Yes, this is a good movie.
It's a fantastic movie.